In a Copenhagen lab, scientists crushed a 3,000-year-old bone and ran a disease test. The result came back positive for typhoid fever. That should have been impossible. Typhoid was first named in 1829. The bacterium was isolated in 1880. Medical history placed typhoid in the modern era. The ancient bone disagreed. The skeleton came from a Bronze Age burial site in the Near East 3,000 years ago. No one knew what bacteria were. No one knew what typhoid was. But people were already dying from it. A disease that would kill millions in modern history had been killing quietly for millennia before it had a name. Scientists extracted the pathogen's DNA from the bone's dense inner matrix. They matched it against the modern reference genome for Salmonella typhi. The match was unambiguous. Typhoid was not born in the industrial age. It was far older than any record showed. The first written medical record of typhoid dates to 1829. The disease had been killing people for at least 3,000 years before that. 3,000 years of epidemic. No name, no record, no count of the dead. This is the investigation that changed what we thought we knew about one of history's. To understand what the bones revealed, you first need to understand why typhoid stayed invisible for so long. Typhoid fever is caused by Salmonella enterica serovar typhi. It is one of the few human pathogens with no animal reservoir at all. Salmonella typhi exists only in humans and spreads only between humans. Wherever typhoid appears in the ancient record, humans brought it there, and humans paid the price. The disease travels through contaminated food and water, silently. Before anyone knows the source is infected, a single carrier can contaminate an entire water supply while appearing completely healthy for weeks. The incubation period runs 1 to 3 weeks of invisible, accelerating spread. By the time the first fever appears, contamination has already spread far beyond the original source. The fever climbs above 40 degrees Celsius. Abdominal pain, a distinctive slowed heartbeat. In severe cases, the intestinal wall perforates and death follows within days. Without treatment, the fatality rate is 10 to 30%. 1 in 3 untreated patients died. That was typhoid's kill rate through all of human history, before antibiotics. Modern medicine did not encounter typhoid as a named disease until the 19th century. For centuries before, it was grouped with other conditions under the catch-all term, continued fever. Typhus, typhoid, and malaria all looked identical at the bedside. No physician could separate them. Pierre Charles Louis distinguished typhoid in 1829 using intestinal lesions visible only during autopsy. Louis gave the condition a name. Nobody asked how long it had been killing before. Carl Joseph Eberth isolated the responsible bacterium in 1880 from the spleens of deceased patients. For the next century, medicine assumed typhoid was primarily a disease of industrialization and urban crowding. That assumption was built on zero ancient evidence. Nobody had thought to look for it further back. The standard narrative was confident. Typhoid was a modern problem born of cities and contaminated. History pointed to the Civil War, the Franco-Prussian War. Typhoid killed more Union soldiers than combat did. This reinforced the modernity assumption. Dense military camps, poor sanitation. A modern disease for modern warfare. The case of Typhoid Mary added to the picture, a cook in turn of the century New York. Typhoid felt like a disease of the industrial age, of cities, and because it felt modern, nobody looked back further than the 19th century for its origins. The laboratory that found ancient typhoid DNA was not looking for typhoid specifically. That matters for understanding the result. The team was conducting a broad survey of pathogen DNA in skeletal remains from near. The method was metanomics, sequencing all DNA in a sample without preselecting any target. Instead of asking, "Is typhoid here?" the team asked, "What is here?" and let the metanomics reads every DNA fragment in a sample and identifies each one against known reference databases. It is the forensic equivalent of running every fingerprint in a room. The bones had been in a museum archive for decades. Skeletal markers placed the individual in early adulthood, late 20s to early 30s at death. Cause of death had never been established. The excavation records listed it simply as unknown. The team extracted DNA from the petrous bone, the dense pyramid of bone behind. From 3,000-year-old material, human DNA survives in short, degraded, incomplete fragments. Bacterial DNA is rarer still. It degrades faster and is far more vulnerable too. A single modern bacterium on a researcher's glove can overwhelm an ancient bacterial signal Every precaution was taken. Full protective suits, filtered air, UV sterilization before and after every step. Every reagent was checked against a contamination database to exclude any modern salmonella introduced during processing. The extraction yielded short DNA fragments, expected for material this old. Metagenomic assembly overlaps short fragments like a jigsaw, building longer sequences from dozens of overlapping pieces. The assembled sequences were mapped against the reference database. Salmonella enterica. A significant fraction of bacterial fragments were aligning to the Salmonella reference genome. The team ran the alignment again with stricter parameters to eliminate any possible contamination signal. The Salmonella signal held. The fragments were too short and too damaged to be any form of modern contamination. The genus Salmonella contains thousands of serovars. Most cause ordinary food poisoning and live freely. To identify the specific serovar, the team compared assembled fragments against a panel of Salmonella. The key diagnostic genes for Salmonella typhi, the genes that specifically cause typhoid fever. The ancient bacterium carried the V inogen genes, the defining molecular signature of S. typhoi. This was typhoid fever. The researcher cross-checked using a second method, mapping the fragments against the full S. Coverage was not perfect. 3,000 years of degradation left gaps. The team also checked whether the DNA showed damage patterns found exclusively in genuinely ancient material. Ancient DNA shows cytosine converting to uracil at fragment ends, a decay signature no modern contamination can replicate. The ancient Salmonella fragments showed exactly this pattern. Classic ancient DNA damage. This was not a false positive. Real Salmonella typhi DNA. The individual in that burial had almost certainly died of typhoid. A disease, not. The team held the finding for 2 weeks before writing it up. Not from doubt, from understanding what it meant. Typhoid was not a disease of industrialization. It had been killing since the Bronze Age, possibly much longer. Continued fever, summer fever, bilious fever, putrid fever. Dozens of names, all describing the same pathogen. For at least 3,000 years, the same bacterium did the same thing. Unseen, unnamed, and consistently lethal. How much of recorded history was silently shaped by a disease nobody could see clearly? How many ancient fever epidemics were typhoid outbreaks, recorded under the wrong label? How many rulers, armies, and populations were destroyed by a pathogen history always identified incorrectly? The ancient bone had opened a door that modern science had never thought to look behind. Before this discovery, other labs had found related signals. Enteric fever pathogens older than any textbook had, and one prior finding pointed at a far more famous catastrophe than any museum archive. The most consequential prior finding came from the oldest detailed epidemic description in all of recorded history. Athens, 430 BC, the city at the peak of the Peloponnesian War against Sparta. The historian Thucydides survived the epidemic and wrote the most precise ancient clinical account of. He described the onset: intense heat in the head, burning eyes, sneezing, hoarseness, violent coughing, vomiting. Then the abdominal phase, spasms, severe diarrhea, a rash of red pustules, extreme thirst, and deep restlessness. The epidemic killed roughly a quarter of the Athenian population. For 25 centuries, scholars debated what the Athens plague actually was. No consensus had been reached. Plague, smallpox, typhus, and hemorrhagic fever were all proposed. None had been confirmed by hard evidence. In 2006, a University of Athens team published ancient DNA results from a Kerameikos cemetery burial pit. The pit held 91 individuals in stacked layers. Emergency mass burial during a catastrophic. The team screened dental pulp from all 91 individuals for every major pathogen that had. Their result? Sequences consistent with Salmonella enterica, serovar typhi, detected in 3 of the specimens tested. If confirmed, the Athens plague, one of history's most consequential epidemics, was a typhoid outbreak all along. The finding was immediately contested. Researchers questioned the methodology, sample size. The debate has not fully resolved. The Athens plague may have been typhoid, but uncertain is a very different conclusion from impossible. The Athens case changed the frame. Ancient typhoid could exist in the historical record. The next confirmed ancient enteric fever case came from Norway. Medieval Norway. Viking Age. Archaeologists at a medieval church site at Viking, in western Norway, uncovered a young adult. A team from the University of Bergen extracted dental pulp and ran metagenomic sequencing on. They found Salmonella paratyphi, a closely related pathogen, causing a clinically identical disease called paratyphoid fever. Paratyphoid C produces the same symptoms, the same intestinal perforation risk. The two diseases were clinically indistinguishable until the bacterium was typed in the early 20th century. For the medieval physician, this was simply a deadly fever. The Norwegian case was the first confirmed enteric fever finding in European ancient DNA, peer-reviewed and damage authenticated. The individual at Myking had died of paratyphoid fever around 1200 CE. The Norwegian case also revised the theoretical model. Medieval Myking was not a crowded industrial city. It was a small agricultural community in western Norway. The disease could persist in small rural populations for centuries, killing quietly, recorded only as an unnamed fever death. Typhoid and its relatives killed in the margins of history. The pattern becoming clear was not the pattern any textbook had described. Typhoid was not born in the industrial city. Researchers turned to the next question. When did Salmonella typhi first become a human pathogen? To answer that, they turned to phylogenetics, reconstructing evolutionary history by comparing the genomes of modern S. typhi strains. Every organism's genome carries its evolutionary past. Mutations accumulate at known rates, creating a molecular clock. By comparing hundreds of modern S. typhi strains from around the world, that tree has a root, the common ancestor of all modern S. typhi lineages. The molecular clock places the common ancestor of all SS. typhi lineages between 2 and 10,000 years ago. Even the most conservative estimate, 2,000 years, puts typhoid's origin well before. The mid-range estimate of around 3,000 years places its emergence in the late Bronze. S. typhi had been evolving and spreading for millennia before any physician saw it clearly. Because S. typhi has no animal reservoir, it was sustained entirely by continuous human-to-human transmission across all those years. In every century since Salmonella typhi first emerged, infected people were maintaining the transmission chain. Contaminated wells, shared water sources, river crossings where sewage and drinking water met. Wherever people gathered in sufficient numbers with insufficient sanitation, Salmonella typhi could establish itself and persist. This is not a disease of industrialization. It is a disease of human proximity as old as civilization. The phylogenetic tree also asked where S. typhi first emerged geographically. The oldest lineages cluster there. The probable origin lies in the ancient riverine civilizations of the Indus Valley civilization. At its height, had tens of thousands living in planned cities. Those water systems were sophisticated but not sealed. Wastewater and drinking water could interact at multiple infrastructure points. A single asymptomatic carrier contaminating a shared well in a city of 30,000 could. There is no written record of typhoid in the Indus Valley. Their script remains undecoded. What exists is genetic evidence, placing typhoid's family tree roots in exactly this region. From South Asia, the phylogenetic data shows S. typhi spreading westward along ancient trade routes and with migrating populations. By the time the Near East bones were buried 3,000 years ago, it was already a disease of the ancient Mediterranean world. Of trade routes, it moved with people because it could only survive in people. 2,500 years ago, typhoid may have struck the most consequential ancient city at the worst moment. The Athens plague ran from 430 to 426 BC, 4 years. The military consequences were immediate. The Syracusan expedition devastated. The empire's expansion halted permanently. The political consequences were permanent. Athens never recovered its political coherence after Pericles died. One epidemic, possibly typhoid, may have permanently altered the trajectory of Western civilization. The Athens DNA evidence remains contested. The researchers did not claim certainty, only that. What can be stated with certainty is the broader pattern enteric fever pathogens have been. The evidence comes from 3 independent sources: ancient DNA from specific bones, phylogenetic dating. Each source is imperfect alone. Together they build a convergent case for revising the textbook timeline. The revision matters because it changes the disease's relationship to human civilization as a whole. Typhoid has been part of the human story for longer than any written record. Every unexplained fever death in the ancient record is a potential typhoid case that will. The ancient Near East bone is one of the few cold cases actually opened. 3,000 years, one diagnosis, but the finding also raised a broader question. How many other ancient diseases have we? Metanomics of ancient remains is a relatively new field. Since then, ancient Yersinia pestis, the plague bacterium, has been found in European. That placed plague in the record 1,500 years before the first written account. Typhoid follows the same pattern. Ancient Mycobacterium tuberculosis has been found in remains dating to at least 9,000 BC, and ancient hepatitis B virus has been recovered from remains spanning 5,000 years of human. In every case, a disease assumed historically recent was found to be far older than. Ancient disease history is systematically underestimated. The record only names diseases after someone learns to distinguish them from all others. For most of human history, the naming came thousands of years after the killing had. The disease was there. The name was not. The researchers describe the finding as opening the invisible epidemic history of the ancient world. That phrase matters. The epidemic history we have was written almost entirely by literate observers in documented societies. But most of human history was not lived in documented societies. Those communities were not exempt from typhoid fever. The ancient bone gave voice to one of those deaths, a death silent for. The practical challenge of ancient pathogen work is considerable. Bacterial DNA is far rarer than human DNA in ancient remains. Of all ancient burials that might contain pathogen DNA, of those, only a small fraction will ever be excavated and tested with metagenomic methods. The findings almost certainly underestimate both the age and the geographic distribution of ancient typhoid. If typhoid DNA survived in one bone, it almost certainly existed at dozens of untested sites in the same period. The phylogenetic clock is clear. S. typhi did not emerge in the Victorian era. It emerged in deep antiquity. The 19th century was simply the first time anyone looked. That distinction has direct consequences for how we think about this disease's future. Typhoid has developed resistance to multiple antibiotic classes. Extensively drug-resistant typhoid emerged in Pakistan in 2016 and has been spreading. XDR typhoid is simultaneously resistant to 5 major classes of antibiotics. Every drug that, a disease that has been killing humans for 3,000 years, has now countered the resistance came from a mobile plasmid, a small loop of transferable DNA carrying. That plasmid can transfer to other bacteria. The resistance is mobile, has spread, and is not confined to S. typhi. The ancient history of S. typhi is directly relevant to this modern problem, because a pathogen that has sustained itself as an exclusively human disease for at least 3. It survived antibiotic introduction by acquiring resistance. Every adaptation that made S. typhi successful for 3,000 years as a pre-modern pathogen is still fully operational today. There are currently an estimated 11 to 20 million cases of typhoid fever per year globally. Approximately 110,000 people die from typhoid each year. The geographic distribution of modern typhoid corresponds almost exactly to where populations lack reliable access. Typhoid today is where it has always been, in places where sewage and drinking water have not been reliably separated. The ancient world had no infrastructure to separate the two. The Bronze Age individual died in a world with no name for this disease. They died of it. Others in the same community almost certainly died the same way. Ancient typhoid killed without a name, without a record, without a single surviving document. The history lives in bone. Bone is a more reliable record than ancient text, in certain ways. Texts are burned, lost, and mistranslated. Bone preserves the molecular record of what killed thee. The ancient Near East bone was in the right conditions. Dense sediment, stable temperature, 3,000 years of those conditions, and the pathogen's DNA remained readable, telling a story no document had ever mentioned. The finding was published, peer-reviewed, and accepted by the research community with appropriate acknowledgement of limits that are real. One individual, one site, one time point. What it does is shift the null hypothesis. Ancient typhoid is no longer a question. Answering that requires testing hundreds more skeletal assemblages across multiple geographic regions and time periods. Work currently underway. Research groups in multiple countries are conducting systematic metagenomic surveys of ancient bone collections. The results accumulate slowly because the work is technically demanding, expensive, but the early findings are consistent. Ancient disease history is older and more continuous than. The most striking implication is what the Bronze Age finding means for every ancient fever epidemic we have never resolved. Ancient sources are full of unresolved epidemics. Livy describes multiple events during the Roman Republic: the Antonine Plague, which devastated the Roman Empire from 165 to 180 CE. The Justinianic Plague of the 6th century CE was confirmed as bubonic plague by ancient. Confirming why pestis in the Justinianic Plague proved ancient DNA could resolve historical epidemic identities. Typhoid now belongs there. Every major ancient epidemic not yet resolved by ancient DNA testing is a potential typhoid. The ancient world had the conditions for typhoid. Everywhere it had settled populations shared. The disease was a plausible participant in any epidemic, at any time. The researchers were careful not to overclaim. One bone proves one case. Evidence must be read at its actual scale, but one confirmed case is enough to change the framework. Ancient physicians were not incompetent for failing to identify typhoid. Typhoid symptoms overlap significantly with those of many other severe febrile illnesses. The bacterium is invisible to the naked eye, lives in clean-looking water, spreads through contact that leaves no visible trace. Hippocrates, writing in the 5th century BC, described prolonged, intense, often fatal ardent fevers. Galen, writing in the 2nd century CE, described a disease with abdominal symptoms, skin rash. Neither Hippocrates nor Galen had any concept of bacterial infection. What they described may now be evidence of S. typhi in the ancient Mediterranean. Not conclusive, but newly plausible. The historian's toolkit for ancient disease has always been limited to texts and physical remains. That tool is fragmentary, expensive, and available for only a tiny fraction of the ancient. But it is real, authenticatable, and unable to be mistranslated. In the Near East bone it matched. That match carries more weight than 1,000 ancient descriptions of continued fever. Before the DNA finding, the story of that Bronze Age death was an unknown fever. After the finding, typhoid fever killed this person. The same pathogen that killed hundreds. The death did not change. The name changed. Ancient DNA does what no other method can: give specific names to deaths that ancient observers could only suffer. The researchers describe the individual in that burial as the earliest confirmed typhoid victim not the first person who ever died of typhoid. The first person who ever died of typhoid died far earlier, in bones. The molecular clock, places S. typhi in the human population for at least 2,000 years, probably considerably longer. The gap between that theoretical estimate and the first confirmed ancient DNA case that gap will narrow as more sites are tested. And as technology becomes more sensitive, future technology will find things in the same bones that current methods cannot yet recover. The ancient record of typhoid will lengthen as the tools improve. What exists now is the beginning. What the beginning already tells us is sufficient to fundamentally revise the history of this disease. Typhoid fever is not a modern disease that emerged with Victorian urban crowding and industrial. It is an ancient disease that has accompanied human civilization for as long as civilization. Every ancient city had those conditions: shared water, proximity, and no way to separate sewage from food and drink. The disease did not need the Victorian era. It needed only human populations. By the time anyone could see it clearly enough to name it. 3,000 years of deaths recorded as nothing, as fever, as miasma. The ancient world had names for disease. It did not have the right name for this one. The Bronze Age individual died of typhoid before the concept of bacteria existed. They died in a world where the only available explanations were religious, humoral, or astrological. None could prevent the contamination of a water source. The ancient world was defenseless against typhoid, not from lack of intelligence, but from the genuine invisibility of its mechanism. The bacterium is measured in micrometers. It lives in water that looks clean. The only defense that worked was separating drinking water from human waste, which ancient Rome's aqueduct system reduced typhoid transmission, but did not eliminate it. Athens had public water systems. Contaminating those wells in 430 BC may have created a citywide catastrophe. The mechanics of how typhoid would have spread in ancient Athens are identical to the contaminate the water, run the 3-week incubation, and by the time the fever begins that sequence, invisible contamination silent incubation, simultaneous outbreak, is why typhoid has always. The Bronze Age individual was part of a transmission chain, connected to infected people and a source they never identified. The rest of that chain is lost. The other cases from the same outbreak will. What remains is one bone, one positive test, one unambiguous result telling us the disease. That single data point changes the conversation about typhoid's history. The full history of S. typhi in the human population is almost certainly unrecoverable. Too much has been lost, but the fragment that can be recovered through ancient DNA, phylogenetics, a story of a pathogen present in human civilization, from its earliest complex forms, a story that medicine missed completely because it was not looking far enough back. The Near East bone, the Norwegian case, the Athens findings, the molecular clock, each limited, each real, together rewriting history, and the rewriting has barely begun. Ancient DNA is being applied to new sites, new time periods. Each application has the potential to extend the confirmed record of typhoid further back than. The next confirmed ancient typhoid case may come from Egypt, Mesopotamia, the Indus Valley, or Bronze Age China. What that evidence will look like when the record has been extended as far as. But the direction is clear. The disease is older than the record. Typhoid fever has been killing humans for at least 3,000 years before anyone wrote. The Near East bone is one message from that long history. It has been read. The message is specific. The individual in that burial had no access to antibiotics, vaccines, water treatment, or germ theory. They died without any of the tools that have made typhoid preventable in the modern world, and in the places today where those tools remain inaccessible. 3,000 years of that failure. It kills 110,000 people every single year today. The ancient history of typhoid is not a story about the past. The researchers who opened that bone were not looking for typhoid, and the DNA answered, typhoid, 3,000 years old. That is how ancient DNA works at its best. Ask what is here. Let the genome answer. Trust the result. The answer was authenticated by the cytosine to uracil damage signature. A small fragment of bone answered a question that had no answer for 3,000 years. The result was a revision to the known history of a disease that kills A. The return on ancient pathogen research is extraordinary. Bones carry answers no other source has preserved across this span. The 3,000-year-old bone that tested positive for typhoid is now a reference point in a single data point that moved the confirmed timeline of a major pathogen back further. There will be more such reference points. The work continues. Each test is a cold case reopened. Each positive result is a death, unnamed for millennia, finally diagnosed. Typhoid has been named, diagnosed, treated, and vaccine, preventable for over a century. The ancient record reminds us how long a pathogen can operate before effective countermeasures exist. The Bronze Age individual died without countermeasures, without a name for what was killing them. They died of typhoid 3,000 years before the disease got its name. Where those conditions persist, people still die. The bone tested positive. The disease was 3,000 years old before any record. That is a strange and remarkable way to learn a piece of history, but it is the only way available. The ancient dead could not speak. Only bone could carry the evidence forward. A dense bone behind an ear, sealed in sediment, waiting for a technology that did. The technology arrived. The evidence was read. The disease was named typhoid fever. 3,000 years before history knew it existed, scientists tested ancient bones for typhoid. The disease was already there. 3,000 years old, unnamed, lethal. The history of typhoid has been rewritten. Not by a rediscovered document, by a fragment of damaged bacterial DNA from a museum archive bone. The machine found typhoid. 3,000 years of it. The evidence in that bone said, typhoid was here long before anyone could say those words. And still is. The 3,000-year-old victim of typhoid fever has no name in any surviving record anywhere in. Their cause of death has now been diagnosed with more precision than any ancient physician. The molecular record of their death outlasted their name, their community. That is what bone preserves when conditions are right. Not memory, not name, not story, just raw molecular evidence. What else is waiting in bones not yet tested? In burials not yet excavated? Ancient DNA research is still in its early years. Typhoid is one entry in that catalog. One disease, one bone. There are others still waiting. Other old diseases, other ancient victims, other bones never opened with the right test. Typhoid tested positive at 3,000 years old. The question now is how many other diseases are older than we think? And the bones have the answer. The technology exists to read it.